the PCT today, how it reabsorbs glucose, amino acids, and vitamins. Yesterday we were looking at what happens in the glomerulus, right? And we said there are two things that happen in the glomerulus. Where we had filtration, and we saw how filtration will take place in the glomerulus, where we talked about afferent arteriole being wider in lumen than efferent arteriole. This will increase pressure in the capillaries, causing small molecules of about or below 900 molecular mass to filter through the basement membrane. We said between the blood and the lumen of the Bowman's capsule, we have two layers. You have the first layer of the capillary endothelium, and in between them we have the basement membrane that is made up of the protein molecules, the collagen and glycoproteins. Then we have the podocyte cells. So then here we have the lumen. So we saw that only small molecules that are below 6,000, 69,000 molecular mass can pass through. And the pressure due to the size of the afferent and efferent was the one causing that to filter through. When you compare the flow rate of this, when you talk about blood proteins not being too large to pass through, they are blocked or they are filtered. We also look at the flow rate, and we said the flow rate in the the glomerulus is controlled by the pressure. So in the blood, we have more solids, fine. So it has a lower water potential. So there's a of water moving back into the blood plasma. But because of the pressure being created because of these two, it oversees the solid potential and water will move always all the lumen, right? Now today we just advance it kidogo and see what happens. What happens in the PCT? For us to go ahead and see what happens in the PCT, yes? High pressure, high water potential.
In the same cell?
Yeah. Pressure will increase water potential. So when there's high pressure, there will be high water potential. Water will move out of that solution to where there's low water potential.
So how about the lumen?
Huh?
You said the water will move out of that
of the plasma.
Oh, of the plasma.
Into the lumen. Yeah. Plasma, blood. Yeah, in the capillary. Out. Yeah. Now, we have the PCT here where we are having selective reabsorption. And the one that is using here is selective reabsorption. That means you're only selecting, not everything. We are selecting what to be reabsorbed. Majorly we have glucose, amino acids, and for me another one, vitamins, and salts. Okay? So before we look at how they are reabsorbed, the first thing we think about is the adaptation of the PCT. We want to learn PCT to its function. The function of the PCT is for reabsorption. Okay? Now we look at the features of the cells lining the PCT. And the first one was they contain microvilli. Microvilli provide large surface area for reabsorption. How? It will give us more co-transport proteins or more channels through which ions can diffuse down. Then we had number 2, tight junctions. In between the cells, they are tightly joined together so that nothing can seep or leak in between the cells. Whatever has to go must pass through the cells, nothing in between the cells. Like if you're having two cells here, or let me draw three cells here. You find that these cells have some space in between. So if there's no tight junction here, some substances can leak in between the cells. But everything has to must move through cells. So in between cells here, we have what you're calling tight junctions. Like when the cells are supposed to be meeting, there's a tight junction. So whatever comes in must go through the cells, not between the cells. Then we have mitochondria. Mitochondria's function is to do what? Huh? Mitochondria. Huh? To carry out aerobic respiration to provide energy. The energy will be used to pump the sodium ions. So it's carrying out aerobic respiration. Remember, when you're learning about respiration, you said in the cytoplasm we have glycolysis, right? Which can happen without oxygen. But anything that goes into the mitochondria, there must be oxygen. That's what I'm saying. It carries out aerobic respiration to provide energy that can be used for active pump of sodium ions. So the other adaptation here we come and talk about sodium pumps. Sodium pumps. Okay? Then we have cold transport proteins. These are the adaptations of the microvilli. We look at each and how they help to allow glucose be transported easily. Actually, the main molecule we are focusing mostly on is glucose. Our body does not afford to lose any amount of glucose. It can lose excess amino acids. We saw it in excretion, where we said when there's enough or excess proteins in our body, we end up having excess amino acid. The body cannot store that. So what the body did, it broke down the amino acid into keto acids and ammonia. Ammonia, which is also combined with carbon dioxide to form urea. Then urea diffused out of the blood, out of the liver into the blood and second to the kidney. And here is where we are now, trying to remove urea from the blood. So, now we move on. I will try to draw a diagram here that can help us maybe see exactly what's happening in the PCT. Now that we have the adaptations, and I have the movement sign here. These are movement of the PCT. Okay? Then I have the cells here. These cells have microvilli. Then these cells have mitochondria. Here we have the blood vessel capillaries. In between these we have the basement membrane. These are our cells. On this side of the cells, we have the sodium pumps. Here's where we have the sodium pumps. So if we look at my cell, actually, it's fully completed. It's talking about all the adaptations. Microvilli with adaptation. Here, in this microvilli, we'll have co-transport proteins. When you say co-transport, it means that it's going to transport more than two substances, or two or more substances, not one. So the first adaptation was microvilli, which is there. The second one is co-transport protein, which is carrying sodium ions and glucose. The third one is mitochondria, provide energy. The fourth one is sodium pumps. And then we have tight junctions. So how is glucose going to move all the way from the lumen into the blood? It starts with the sodium pumps. The first thing we say is the sodium pumps, they pump sodium ions from the cell into the blood against concentration gradient. So we're going to pump them from where they are low to where they are high. Once they get here, the blood carries them away. That's the work of the blood, the plasma. So once they get in the blood vessel, the blood carries them away. But here, we are actively pumping them from where they are low to where they are high, using the energy from respiration. As a result, you find that the sodium ion concentration within the cell here will go down. It lowers the sodium ion concentration. As a result, here from the lumen into the cells, they diffuse down the gradient. So here sodium ion just diffuse from high to low because here we have pumped them, all of them into the blood vessel. So here they just diffuse from where they are high to where they are low, down a gradient, through co-transport protein, meaning that as they are moving down from high to low, they will provide for us energy that will carry for us the glucose molecules. That's why we ask you, how does glucose move from the lumen into the cell? It's by active process. Active in the sense that it's depending on the concentration of sodium ions to diffuse from where they are high to where they are low. Here they are low because we are constantly pumping them into the cell using the sodium potassium pumps. So as they diffuse, here they are diffusing. From the lumen into the cell they are diffusing from high to low. But they are passing through a co-transport protein. Remember when you're learning about cell surface membrane in AS, we said polar molecules cannot cross the cell surface membrane. Reason was, what was the reason? Polar molecules cannot cross the cell surface membrane. What was the reason? Because of the hydrophobic region, it repels all polar molecules. So for any polar molecule to go across the cell membrane, it needs something to carry it. So in this case, sodium ions themselves here are polar ions. They can't cross the cell membrane just freely. They can't diffuse through the cell membrane. Glucose is one soluble. It can also not cross the cell surface membrane. So it needs something to carry it. And you think about carrier protein, which is referring to it as a co-transport protein. Co because it's going to carry for us sodium and. Are you together? Now, once glucose gets into this cell, what happens to the water potential? Once glucose diffuses from this cell from the lumen into the cell, what happens to the water potential? It lowers water potential, right? So by osmosis now, water will follow. By osmosis, water is now following because we have now a lower water potential within the cell. Then glucose will come here because they will also now go through transport proteins. But now so meaning that within this layer here, actually we also have microvilli this side. We have microvilli both sides, this side and this side. So here we also have glucose transport proteins. So once glucose diffuses here, it increases the concentration, they now diffuse down into the blood. Here they are just diffusing. But here they are depending on sodium ions. The more the sodium ions they are, they will carry with them the glucose and amino acids and vitamins. Point at home? Is it clear? Any question for me? Yeah?